goal of this week's lab is to examine cells. We're going to use the microscope to examine cells, and we'll be looking at various microscopic techniques to examine a variety of different cells. This is the microscope we'll be using. This is a Brightfield microscope. Brightfield microscopes begin at the bottom with a light source. The light source shines light up through the substage. There's a condenser that basically focuses light. The light passes through an object, then goes directly through. It's transmitted up into one of these structures, which are called objectives. It then goes through this tube, bounces off a mirror, hits the ocular, and it's magnified again by this lens, which is a 10x ocular. Here we have various objectives. We have a 4x objective now in place. We always want to start with our lowest magnification in place because then it's easy to move things on and off the stage. We have a 10x objective, a 40x objective here, a 40x objective here for something called phase contrast, which we're not going to look at, and a 100x objective up here. Okay, so our 4x objective is in place. If we have a 10x ocular, a 4x objective, 4 times 10 is 40x. That will magnify a slide by 40x times. Our 100x then is our maximum. Our 100x is designed to be used with oil. We will use the 100x at the end when we look at bacteria. That 100x then times the 10x ocular is 1000x. Now we also have a camera attached. This camera then goes right into the eyepiece. So we basically take this eyepiece out, put this camera in, and we have it hooked up to a computer. We can get a live image of the computer. So everything that I see in the microscope, you will see on this computer image, and you will get a nice live view of exactly what's going on with the microscope. For example, here's a slide that I made up. This will be one of the slides that we'll be looking at later in lab. We'll just throw it in here really fast so that you can see what's going on. And we'll focus it, kind of move it down a little bit, put some stuff in the center that looks cool. Then what I can do is easily switch to a higher magnification, focus it. Okay. Center the object. There's actually several things on here which are pretty neat. I just have to choose one. Center it, focus. Alright, then we can center it again and look at various objects. So here's one critter that we'll actually be looking at and examining a little bit later in this lab. The first technique I will show you is how to make a wet mount. So we have a culture here. This culture is of mixed amoeba. The first cells we're going to look at are amoeboid cells, which are really cool cells. We have cover slips here and a microscope slide that has all been cleaned off here. So I'm going to take the lid off of this. Key thing about an amoeba slide is that the amoeba are all in the bottom. They're heterotrophic, so they're feeding on all of the debris. So what I want to do is I want to make sure that I go down into the bottom here and gently suck up all the debris I possibly can. So you can see all that debris in there. And then take this, put drops on here. Okay, we'll close this lid off. We'll leave it a little bit open because these actually have to have air, so we don't want to close it completely. And I will go in and place a cover slip on the slide and prepare it and have it all ready to go. This is our amoeba slide. We're right now under the 4x objective, so it's magnified 40x times. Amoebas are interesting. They're single-celled organisms, and they're actually quite large single-celled organisms, and they're very slow-moving. The key to finding an amoeba is knowing what you're looking for. Okay, in here there are actually two different types of amoebas on this particular screen. Amoebas move slowly, and they expand out with long finger-like projections called pseudopodia. So I'm gently going to move into the very center, the amoeba. Okay, and I know it's an amoeba mainly because I know what I'm looking for. And you can see other things moving all around. The amoeba actually is not moving very fast. I'm going to take the 4x, I'm going to shoot up to the 10x, focus it. Okay, and there's our amoeba. We'll move our amoeba right into the center. And amoebas are very interesting in pond water because in pond water you have, all of your various levels of an ecosystem. Okay, you have your producers, which then are photosynthesizers. You have your consumers, which then are going to eat the producers. And just like in any ecosystem, we have our predators. So these big giant amoeba are actually predators, even though they move extremely slowly. Okay, if you've ever seen the movie The Blob is a giant amoeba that goes in and eats people. These amoeba then are eating what are called euglena. All these little guys swimming around are euglena. And what the amoeba does is that it tries to trap a euglena inside its long legs that it's moving around. Okay, those long legs then are called pseudopodia. And you can see right here that the amoeba is engulfing these little green guys right here. So they basically swim in. The amoeba then attracts it in, surrounds it with its pseudopodia, and then ends up taking it into the cell. Inside this amoeba cell, you can see all kinds of vacuoles inside where it has organisms that it's captured and it's going to eat. Some of the interesting questions I always have about an amoeba is what makes it go the direction that it goes. So we can see that this amoeba is just moving along. It's moving forward from left to right. And I really don't know what clues or chemical signals it takes then to actually figure out where to move. But they definitely have certain personalities and will move in various directions. Okay, and I'll just let you kind of view this amoeba here as it's moving out. You can see that nice euglena there swimming around at the top of that layer. That amoeba is definitely attaching its pseudopodium on that layer there. We can move down and about and see. It's interesting because it obviously didn't like this little greenery to the left. So the back left, it left that. It decided not to eat that at all. And instead, it looks like it's pursuing that euglena right there. So see that euglena right at the tip that it seems to be trying to grab. Okay, we'll kind of center it on that euglena. We'll see who wins, the euglena or the amoeba. That euglena kind of is staying there. It's not leaving. It's hanging out right where the pseudopodia are. Oop, the euglena left. So this particular amoeba did not get that euglena. Okay, but there's a lot of euglena around. You can see the various euglena that it's already eaten are inside and being digested in the cells. All right, so this is a single-celled organism. It's a protozoan then, and it is an amoeba. Later in the semester, we will be looking at cells in people in the immune system, which are called amoeboid cells and move also through amoeboid movement. Now the next amount I want to make is of what are called ciliates. Ciliates are different than amoeba in that they move by cilia rather than pseudopodia, and we'll examine that under the microscope. So here's my culture. This is of mixed ciliates. Again, it's from Ward's Biological Supply Company. It has a lot of ciliates inside. I'm going to turn it around this time so you can actually see all the debris in the bottom. These guys, again, are heterotrophic, so they're hanging out in the bottom. And what I want to do is I want to get in and carefully suck up the muck off the bottom. I'm actually going to drop a few drops out so I really get the muck when I put it on this slide. A little bit more here. Okay, There we go. Again, I'll put the lid on. Don't totally screw it on. And we'll get a cover slip. You have to be careful that you don't get more than one cover slip. Mistake a lot of students make is by getting more than one cover slip. There we go. And our wet mount is ready to be viewed with the microscope. All right, here is our view of the ciliate culture under the 4x objective. And we can see a lot of ciliates. So all of these guys zooming around then are different types of ciliates. You can see the long, dark, worm-like ones here. These two long worm-like guys there, those are Spirostomum. The rose-colored ones in here are called Blepharisma. You can see a lot of Blepharisma in there. The ones that are the same size as the rose-colored ones that are not rose-colored, those are Parameciums. You can see a nice Paramecium there in the center. See the rose-colored one being the Blepharisma there. Then you have smaller ones, which are Colpidium and various other guys. Okay, you can see that amoeba directly in the center there. All right, if we focused in on that there, that guy's an amoeba. So we have all kinds of things on this particular culture. Okay. All right, so I'm going to search around a little bit and then find some things to try to focus in on so you can actually see the cilia. There are certain types of ciliates that don't move very much, and they attach themselves to basically pieces of debris. There's one of these right in the center. It's called a Vorticella. Vorticella are pretty cool because they have a stalk. They'll attach to various devices, but they're still going to act like a normal ciliate. Now, as I mentioned last time with the amoeba is a predator. As a predator, then the amoeba will capture live organisms and eat those live organisms. These guys then are more debris eaters. So right in the middle here, this is our Vorticella. And we can see that it has a stalk and it's just sitting there. And we can get really close to it here. There it is. And right around its mouth, you can see it has a mouth. Around its mouth, it has cilia. Those cilia then basically create a current, and then it uses that current to suck in all of the debris around the outside. So basically sucks that in. It's a filter feeder. You can see a lot of various cells, small cells and things inside the Vorticella itself. So the Vorticella is a single-celled organism. It's larger than a lot of the other cells that it's eating, but it is much smaller than the amoeba. These guys, I want to focus in on this guy, the greenish guy with the super long stalk there. Okay, it's a single-celled organisms. All of these are single-celled organisms. That means they're one cell that make them up. There are other things in here that we'll look at in a minute that are multi-celled organisms that occupy a very similar niche to these guys. But this guy is a stentor. He's a ciliate. You can see that ciliate around its mouth. Okay, we'll just focus in. There's a Vorticella next to it that we looked at earlier. And it basically is similar to a Vorticella, only a lot bigger and eats a lot bigger stuff. And we can see it here very well. You can see all that cilia around its mouth. Then the stentor then will beat its cilia and filter feed. Okay, here's another guy. This guy's kind of interesting. It's an amoeba-like organism called an Actinospherium. The Actinospherium there's in dead center. It's right next to a ciliate, which is a Blepharisma. And the interesting thing about an Actinospherium is that it is a predator, like other amoebas. And it has these long, spiky structures sticking out, which are its pseudopodia. The pseudopodia then basically act to capture prey. And so they're sticky, and so when a ciliate like the blepharisma comes in next to this actinosferum, the long pseudopodia then basically stick to the blepharisma, and the blepharisma cannot escape. So here we can see the blepharisma. The blepharisma is a ciliate. You can see that it's beating its cilia, creating currents. So the blepharisma can still feed, but it can't get away. So the pseudopodia are sticking to the blepharisma, and the blepharisma is unable to escape. So if we go up under higher magnification, we can see the mouth of the blepharisma. We can see the cilia all around the blepharisma, but you can also see how those pseudopodia are attached to the blepharisma, and so it's kind of stuck there and can't get away at all. Okay, so then the actinosferum, then, if it wants to eat it, it will slowly kind of hang on to this guy, not allow it to escape, and then eventually it's going to pull it in slowly, drag it in, bring it inside, and create a food vacuole and eat it. Okay, a lot of literature showing that actinosferum eat quite a few of these blepharisma. Everything we've been looking at is unicellular, but right here we have a multicelled organism, which is the exact same size as the unicelled organisms. And this one has about 100 cells, and it's called a rotifer. Go up under higher magnification. Even though it's multicellular, it's easy to identify because it inches along. Okay, it actually has cilia along the top, but those are all teeny tiny cells. It's about 100 cells versus the single cells of the big guys. It has a full crop inside, has a full digestive system. It beats its cilia, uses its cilia much in the same way that a paramecium or a stentor or bordetella uses its cilia. So see that cilia around the top, see that crop actually eating food. So it kind of chews its food and eats it and then digests it more like us rather than like, oh, there's another one. So even though it has the cells are extremely different, but they have come together in a multicelled organism to occupy a very similar niche to that of a paramecium. Next, we want to look at human cheek cells. So in order to look at human cheek cells, I have a variety of items. I have a microscope slide. I have toothpicks that I'm going to use to get the cheek cells off of my cheek. I have a stain called methylene blue. I then have a staining tray that we're going to use to stain the slide, and I have a Bunsen burner to do what's called heat fixing the slide. So the first step is to basically take a drop of water. We'll put a little teeny tiny drop of water on the slide. I'm then going to grab a toothpick. I'm going to rub it on the inside of my head. So all on the inside of my cheeks, I get all kinds of cheek cells on that toothpick. Now I'm just going to then take the toothpick, circle around a nice big circle there so that it quickly evaporates on that slide. Okay. And as it's evaporating there, I'm going to just set it up here on the staining tray. We'll move this wash bottle out of the way there. Now that my slide is dry, it's time to do what's called heat fixation. Heat fixation is basically taking the Bunsen burner, allowing heat to go on the bottom of the slide so that the cells stick to the slide. Later on, I'm going to add a little bit of methylene blue, then we're going to wash it off with a wash bottle, and we want to make sure that the cells don't slide off. So to start with, I'm going to light the Bunsen burner. Okay, we'll get a nice flame going. You see that nice cone on that Bunsen burner there. I'm now going to take my slide. I'm going to just go over slowly twice over this slide, counting 1 Mississippi each time. I'm going to angle the slide away so when the flame hits it, it goes that way. You know, a lot of people use clips so that they are a lot farther away from their fingers. So we're just going to go 1 Mississippi, 1 Mississippi. And put that there, and then we'll be set. I'm going to take my methylene blue and cover the slide with methylene blue. And cover the smear with methylene blue. Alright. So let's smear that. We'll get covered with methylene blue. We're then going to wait for a minute. And the methylene blue has been on for 1 minute, so I'm going to wash off the stain. So I'll just take my wash bottle, my slide, angle it down, and just simply wash all of that methylene blue off. By heat fixing over the heater that my cells stayed on when I washed it off. Now all I have to do to finish this off is just basically blot it dry with Kim wipes, and then I can look at it underneath the microscope. Okay, now we want to look at the cheek cells. So here's a view under the 4x magnification of the slide that we just made with the cheek cells. These cells are stained with methylene blue. We can see little itty bitty teeny tiny cells in there, and each of them have a little black dot. That little black dot is the nucleus. I'm going to shoot up under here the 10x. We can see them a little bit better. Okay. Then I'm going to go up here under the 25x. It's a little bit better. Finally, then we'll move up to the 40x. Increase the light to increase the resolution. And focus so we can see these cheek cells under the 40x objective. Actually, we can see them really clearly here. So there's a nice cheek cell here. The little blue dots are actually bacteria on the cheek cell. We're going to look at those in more detail. If I can move this gently, and we can see that cell right there. Okay. Now what I want to do is if I'm going to go up under oil to look at this, then I need to get that exactly in the center. So there's our cheek cell. It's under our 400x magnification. We're now ready to move to the next step, which is going under oil. To go under oil on the microscope, what I'm going to do is I'm going to swivel halfway between my 40x and my 100x, put a drop of oil directly on the microscope slide, then swivel in my 100x. This is my oil. I'll open my bottle, swivel this halfway between those two, and then put a drop right on the microscope slide itself. Now I'm going to swivel into 100x. Once I'm under oil, I can never go back to the 40x because this is not made for oil, whereas the 100x is. Oil is designed to make more light. As you increase magnification, you need more light to increase the resolution to actually be able to see a cell. So we need that oil on there to have enough light to visualize those cells underneath the scope. Okay, so here's our cheek cell under the 40x objective. I'm going to take it under oil, so I put it halfway between the 40 and the 100x objective. I'm adding a drop of oil to the slide. And then I'm going to wheel in the 100x objective into that oil. Very, very, very gently focus. Okay. And not doing too well. And getting the focus, there we go. I have to slow down on that focus. To be able to get it. Beautiful. There we go. Nice. So the bigger blue dots then are the nuclei. So we can see 1, 2, 3 nuclei in this image. So there's 3 cheek cells kind of on top of each other. Then you can see all those nice little blue dots. Those are the bacteria in the sample. So a lot of bacteria on these cheek cells. And you can see the, oh man, my bad. So we can see all these tissues. So I gouged my tissue really well. And then we can get those bacteria. So bacteria have a variety of different shapes. you can see a lot of different shapes in here. You can see most of them look like very small rod shapes. A lot of them are in pairs, so you can see how they're in pairs of 2. We have a chain in the upper middle, kind of a string of 1, 2, 3, 4, 5, 6, 7 of them there. We have a couple ones that are a little rounder, which are called cocci, which are rather than rods. Most of these are small rods, but there are a few that are a little bit coccus shaped. So a variety of different ones. You can see how big the nucleus is, right? And then a cheek cell is a lot bigger than the nucleus. So bacteria, you can see how tiny they are compared to the eukaryotic cheek cells in our mouths.